So hello and welcome to our Drug Hunter Flash Talk today. I'm your host, Dennis Koester, and we're really happy you made time to dial in today. We will be talking about mechanistic ADME with Fabio Broccatelli today. So good morning, Fabio. It's good to have you here.
Great. Thank you for the invite, Dennis, and good morning to you and to everyone.
And now, without further ado, I want to introduce our guest, Fabio Broccatelli. Fabio is currently the head of DMPK at computational chemistry at Altos Labs, and his PhD and postdoctoral work was conducted at the University of Perugia, UCSF, and the Institute of Cancer Research in London. And he focused on ML predictions of ADME properties, virtual screening, and in vitro to in vivo PK correlations. Previously, Fabio built the computational ADME team within the DMPK department at Genentech and also led the San Diego preclinical DMPK group at BMS, where he supported the oncogenesis portfolio. His main research interests include ML and AI, PBPK, PKPD, and IVIVC across multiple different modalities. Fabio has authored over 37 peer-reviewed articles, patents, and book chapters, and serves as a reviewer on more than 15 scientific journals. He also initiated and co-led the in silico ADME IQ group. Again, Fabio, it's a pleasure to have you here today, and welcome to the Drug Hunter Flash Talks.
Thank you, Dennis, and thank you for the kind introduction.
Awesome. Yeah, and before we get started, in our good old fashion, we're going to ask a few questions to get to know Fabio a little bit better. So Fabio, if you could share with us what inspired you to start a career in drug discovery?
So I was very fortunate during my college and master's years to be in contact with the Professor Kushani's lab, and they were showing how by being thoughtful about how you design experiments and use information in chemical structures, you could actually decrease animal consumption significantly. And that's something I was very passionate about and inspired a lot of the work that I've done through the years, and I still do. I still believe that that's a field that can continue to benefit from the knowledge that we're creating, and I do see that panning out day by day. So I'm very excited about the progress we're making.
Awesome. Yeah, that's a very noble mission, and yeah, you and your work are certainly contributing to that goal. So that's very fulfilling to see. What's your favorite molecule, Fabio, and why?
All right, favorite molecule. I used it as example in papers and presentations. So I'm repetitive that way. It's deutetrabenazine. There was a drug, tetrabenazine, very helpful drug to manage symptoms of Huntington disease, but due to the high Cmax, there were side effects that were actually pretty scary. And simply by understanding where the site of metabolism for the molecule was, researchers were able to substitute hydrogens with deuterium. So no big changes in physicochemical properties, just simply pinpointing the metabolic soft spot. They were able to lower the dose, extend the half-life, lower the Cmax, and have a safer therapy for patients that are in need. So I think that's a really cool story.
Yeah, wonderful example. Thank you so much for sharing the story. What are you most excited about in drug discovery today?
Well, something that gives me excitement and also FOMO, it's all the new modalities. As I started from small molecules, and then I ventured into degraders, and then ADCs, and then more biologics and oligos. So I'm touching a lot of these modalities, and I realize the opportunities are endless, and especially when it comes to combining all of them. So as I go into different conferences, I'm really curious about what people are thinking and what technologies they're developing, because the combination of these different modalities, I think, has endless opportunities. And I think we'll see transformative therapies coming to market in the next few years that exploit this principle. It's hard for a new scientist in drug discovery to keep up with all of this, but it's definitely very exciting.
Yeah, no, I totally agree with you. I couldn't be more excited about the scientific future of drug discovery as a whole. And yeah, as you pointed out, so many different opportunities and so much more different potential that we have in our hands today. But yeah, if you could go back to the start of your career, Fabio, and give yourself some career advice, and maybe give some career advice to our younger audience members here today, what would you say to your younger self or the younger audience members here today?
That's an interesting question. I I don't see myself as a particularly wise researcher, so I don't know that I have great advice. I think what worked for me is really imagining what a future could be for drug discovery and seeing what my role could be within that future, and at the intersection of what are my skills, what is my interest, and what I think is going to be important. Um, I think when I was thinking of in silico ADME maybe 14 years ago, um, my vision wasn't as very well developed as it is now, but it was in the ballpark, and maybe wasn't everybody's view of the field. So I believed in that, and I got the skills to pursue that, and I stayed with it, and for me that worked out. I also think that we gotta be flexible and be able to change and take in from what the field is learning. So at the same time, have a vision, align your skills with that vision, but be ready to flex it and change it. And that worked out for me. I don't know if that's gonna work out for everybody. I may be the only person that in 2022 not worked out, but significantly decreased the amount of time spent in machine learning. I was doing a lot of machine learning in 2022, and everybody was getting into that field. And I thought, I think I'm gonna do more PBPK now. So I don't know that I'm the best example, but that worked out for me.
Great, thank you, Fabio. No, I think that's great advice. I mean, having a vision is, I think, very, very important to see what could be achieved and aligning those skills. I think that's great advice. Last but not least, what do you like most about Drug Hunter?
Well, I don't know how you feel about our day-to-day life, but I'm feeling like there's so much information around me and so much attention grabbing activities, and I can't keep up. And science is becoming a little bit like that with all the journals, all the different LinkedIn posts that are really interesting, but we don't have enough time in a day to learn all of that on top of our working hours. And I like that drug hunters really seek out this information and transform that into knowledge. So that really decreases the time I need to learn while retaining all the benefits. I think I wish there was more of that in any other field of life as well, because I think attention right now is the most valuable good that everybody is after, and the time that you guys get us is very valuable.
Thank you, Fabio. That means a lot to us, and that's greatly appreciated. Greatly appreciate your kind words. Yeah, and before we launch into Fabio's presentation, I wanted to show everybody one little piece of the Drug Hunter platform. As Fabio pointed out, we're trying to distill drug discovery and point out the areas where where there's some things that you can actually apply to your project and knowledge that can actually be gained. So one of the things that we have recently built is this curated patent search here where you can find patents that we distilled down here, and we now release those on a weekly cadence. You can search this by target, therapeutic area, originator, and even you can search in the annotation. So for example, if you're interested in patents that contain PK data, that's oftentimes very difficult to find by other means, you can just type in here in the annotations PK, and now in all of these patents, you will have data disclosed that where you have oral bioavailability in a specific species given for a specific target. You can narrow this down to a given therapeutic area, a target that you're interested in, or an originator. And with that short demo, I will hand off the screen to Fabio. And we're very excited, Fabio, to hear your presentation on mechanistic ADME today. So welcome again to the stage.
Thank you, Dennis. All right. Maybe let me start with what is the definition of mechanistic ADME? Because I feel like in making these slides, I should have had more definitions for the broader audience we are facing today. With mechanistic ADME, what we're talking about is using first principle to derive our models to predict ADME. Very much like physics-based modeling for computational chemistry, where you start from a mechanistic description of the molecule. With mechanistic ADME, you're describing all the processes, and you may be using linear equations, you may be using a bunch of differential equations, but the point is, you know the mechanism. So if you know the mechanism, and you're able to describe what's happening in the animal, you can assume you'll be able to describe what's happening in the human if the description of the animal pans out. And that's very powerful because in the human, for the animals, we may have databases of data, but for the humans, we have very limited databases. So machine learning, it's limited in its ability to predict the human, I feel, because the precedent is fairly limited. But I do think that there is space for machine learning, there is space for in vitro and vivo correlations, and they all can come together in a toolbox. And that's what I'm trying to describe today. And what I'm trying to describe is how we use all of this to assess quality in molecule making and in compound progression. So that obviously has to do with modeling and the science that we describe, but it also has to do with what does the field need? We don't want to be solving a problem that it's not a problem. So I'm going to try to get to that. And I'm going to try to tie decision-making to the sequence of assays, to the assay cascade. Because obviously we make decisions based on the data that we have or the data that we project. So what we acquire first makes a big difference. And we, as medicinal chemists, I do consider myself somewhat of a medicinal chemist. I hope I don't offend anybody by saying that. As medicinal chemists, it's very important that we tie these results to physicochemical properties because that allows our chemical intuition to take action. I'm going to get to that. But I do think that after we come up with ideas, there are better tools these days to score these ideas compared to simply looking at physicochemical properties. So I think physicochemical properties is very good for idea generation. So when it comes to scoring, we can do better. And I'm going to present this concept of mechanistic multi-parameter optimization functions for that. And I'm going to give examples on project work. Okay, so this was 20 years ago. We will be talking about how many failures we have in the clinics because of PK, because of poor PK. But currently, that's no longer the issue. I've seen that if we have a good package for a clinical candidate with all the data, usually we do a good job at predicting PK. And sometimes companies do fail in the clinics, but it's probably because they know they're taking a risk and that let's see if that pans out. But it's rare, very rare that you have completely unexpected PK in development. May happen, but it's not as frequent as it used to be. But still, research is half of our spending, right? Development is half of the spending, research is half of the spending. And research is way less predictable in terms of timelines. And part of the reason why we're successful in development is because we are getting bigger and bigger packages of data in research, and that got expensive. And we don't get all the data immediately. The decisions that we make on the new compounds, it's based on very limited data. So how do we combine these packages of limited in vitro data and large packages of machine learning models? How do we assemble maybe 12, 15 properties into one judgment? I'm going to be talking about that today. So I think a good introduction to the problem is assay cascade. This is something that I didn't have an understanding before getting into industry, but now in industry, I think deciding the assay cascade is one of the most important steps for a project, because you want to have the information that really enables decision making as early as possible at a reasonable time and cost. And you want to leave experiments that characterize the molecule but are more expensive and somewhat more predictable later on, so that you are able to advance the right compounds. We start from hundreds of ideas, thousands if we think about computational approaches to drug discovery. And every week for a project, maybe we select 20 compounds to make. They're very expensive, so compound selection is very important. Typically in those projections, clearance, intrinsic, and potency are the most important properties, and they're also not as predictable as others like permeability and solubility. So we take care of that in our tier 1 assays. And then in the tier 2, typically you counter screen with potential off targets and properties, ADME properties like solubility and permeability that predict the absorption. This may change project by project, of course. For good compounds, we may do a PK study in tier 3 that is expensive, has ethical implications. It's something we want to do for the right compounds. And in parallel, probably we will be collecting DDI data. So we start getting a sense of some clinical facing questions. Compounds emerging from all of these, maybe 10, maybe 15 in a project, maybe 20, depending on how different companies operate. They're tested in more expensive and informative experiments like PK/PD, like alpha experiments. These are longer term. These are in diseased animals. And because we make that investment, probably in parallel, we acquire in vitro tox data, which is also something very important that could kill a molecule potentially. And then for the really good compounds that we consider as potential clinical candidates, we may do high species PK and we complete the package of all the in vitro data across all the functions. So as we keep going down, the number of compounds decreases, the cost increases. You can look at this in a composite manner. You can look at what are the number of molecules I'm going to make in my project and what is the cost for each different tier. You can have assumptions on how many compounds are going to progress. And that's what I tried to do in this slide. This is not general. This is not a real life example. This is my own understanding. People may disagree, but by my own math, the outcome of this is that most of the cost goes into synthesis and tier 1 testing. Because if you make a molecule, you're going to test it in tier 1. So you can see those synthesis in tier 1 as costs that are together. So that's the largest cost for a project. You want to make the right molecule. And then the other spike is deciding what goes in vivo in the first pass PK. So these are the two areas where I think multi-parameter optimization can really help in decision-making. And as we progress, the number of compounds goes down and the quality of the compounds is gonna be dependent on the decisions that we made at that stage. So that's even more emphasizing how important these decision points are. Okay, so how vast is the chemical space that we're going to explore? I took a pet example. This is not realistic. I just simplified my assumptions. I grabbed a molecule from the Drug Hunter webpage, and this is a molecule that I'm familiar with because I used to work at BMS, Celgene, at the Celgene site at BMS, I guess. And what I did for this molecule, I divided that in 4 different areas, 4 different boxes, and I'm saying, let's hypothesize that for each one of these boxes, we made 25 variations. Again, not realistic. We probably made more variations for box A and less variations for box B, but I'm simplifying the calculation here. And I'm also saying, let's assume that for each one of these, I could make 100 more variations. Not realistic. We probably can do 1,000 variations, but keeping it simple. How vast is the space that we could explore with chemistry? Well, we can look at this from different lenses. We could recombine pieces of molecules that we already made. No novel structures, just different combination of past SAR. I'm going to label that as exploitation. We are exploiting knowledge that we already have. In that space, machine learning models are very, very good at predicting potency because they memorize the SAR. There's going to be exceptions, but it's pretty good. It's pretty safe space for predictive modeling. Caveat is, unless you already have all the right groups, you're not going to identify anything that is good enough, right? You need to find the good groups before you rely on that. So that's a good thing to do later on in a project, but not so much in the beginning. So in the beginning, and for most of the project, I think the energies are going to be into proximal exploration. You're changing one group at a time. And you're changing one group at a time because you know that docking, free energy perturbations, whatever approach you use to predict potency, it's flawed and it's gonna fail many times. So by that assumption, you want to know when you're failing, you want to know why you're failing in your prediction. What is the reason? If you start changing multiple groups, you don't know what that is, what the reason is. And our ability to predict potency, it's decent if we change one part of the molecule, but if you start changing multiple parts, it gets lower and lower. So that's why I think this full exploration that many of the machine learning papers talk about these days, I don't think we are there in the field. Problem not being the search algorithm for machine learning, problem being our ability to predict potency. If we start making a lot of those compounds that are inactive, we don't know why they're inactive. So I think as of now, most of the field, for what I've seen, is operating in this exploitation and proximal exploration space. I think as our ability to predict potency would increase, we do more of the latter, and this would be an incredible addition for the efficiency of drug discovery, but we're not quite there yet. I don't think the search algorithms for machine learning are the problem. I think the problem is the ability to predict potency there. Actually, I think reinforcement learning for some problems have been a phenomenal advancement, but that's more for vitro screening at this point rather than medchem campaigns. My bias. I don't know that everybody agrees. All right. So this is a process, and that makes it that's an important introduction, because then we get into how do we generate ideas, and I would say most of the ideas we generate are based on human intuition. And that's where physicochemical properties are really important. If you're a chemist, computational chemist, you want to optimize half-life, you're going to be thinking of, okay, I need to fix my metabolism, but I cannot lower my lipophilicity because I want to maintain the volume and distribution. So a group that can retain lipophilicity and it's not labile, maybe it's CF3, whatever. You connect that objective with chemical intuition through the physicochemical properties. So understanding of that makes really quality hypotheses. And it's most of what we do these days. So efficiency matrices, physicochemical properties, still very, very important. At that point, when we invested in that thought process and we thought about synthesis, and we know that some of these predictive models that we use may fail, we use the models as a complement to our intuition, but we may disregard some of the predictions because we really like our hypothesis. Another approach is full enumeration. You may know a reaction that is viable for your chemical matter. You may have a library of amides. You may couple them. You may enumerate tens of thousands of compounds. At that point, your models are very important. They narrow down to the best hypothesis. And then the human is kind of reviewing what comes out and maybe diversifying, but that's model-driven. All right, so I talked about physicochemical properties. Let's give a little bit more practical sense of what is this problem statement. Why are they important? Lipophilicity in particular. Lipophilicity drives each of the variables that we're optimizing for PK and potency. This is a good example of that. This is a very nice picture from Miller and other people at Merck, a very good paper. They're showing how the free volume, and this is for permeable small molecules where free drug hypothesis applies. It's an important caveat. I don't know that we can say this about bifunctional degraders, but we can say this about most small molecules that are permeable, as long as the transfer between tissues is faster than the elimination from plasma, which is true for most oral drugs. So the free drug, free volume, is highly correlated with lipophilicity. And you see that that correlation is not really skewed. The compounds that have the best dose projection, so the larger squares, they are equally distributed. If you look at the clearance unbound and lipophilicity, you'd see that there is a correlation, not as strong. And you also see that the compounds with the better dose projection are skewed on the right. What that means is that given a clearance value, if you can have that clearance value at higher lipophilicity, you're going to have a better dose projection, generally speaking. Why is that? Well, probably is because your higher lipophilicity is going to increase the volume, and you're going to have comparatively a better half-life. And also, more lipophilic compounds, and we get into the third plot, tend to be more potent. So for potency, you have the opposite trend. You still have a correlation with lipophilicity, but you find that it's skewed. The better dose projection are skewed on the left, which means if for a given potency you manage to reduce lipophilicity, you're probably going to help your clearance with that, and that's going to be good for dose projection. So you change lipophilicity, you're going to change all the properties. There isn't a good lipophilicity range, in my opinion. There's a more workable lipophilicity range for oral drugs, yes, but outside of that lipophilicity range, we have a lot of blockbuster drugs. So it's a matter of how do we make it work and how do we think of this data relative to lipophilicity, and that's where I think lipophilicity metrics are very, very valuable. And I'm going to get to that in a couple of slides. Okay, so traditionally, when the problem statement of dealing with 15 variables became prominent in drug discovery, the first approach that I've seen from MedChem has been these linear MPOs, this progress compared to optimizing for potency and then hoping that everything else works. But I think we can do better. Linear MPOs, essentially you apply a desirability score to each one of the properties you're optimizing, and then you sum them up. What this is missing is that based on the PKPD driver, you want to achieve a high Cmax, high C average, or high C mean. You're going to need different approaches, and this does not cover that. This is also missing that a lot of these variables, you want to look at them as products or ratios. Volume and clearance, it's the product for half-life. Clearance and potency is volume and clearance is the ratio. Clearance and potency is the product for C average coverage. You're missing that here. So how can you deal with that maybe more efficiently? I like the efficiency matrices. These are very helpful when you're optimizing one property at a time. Here, what you do, you normalize your potency by lipophilicity in the case of LLE, or you normalize your metabolic stability by lipophilicity in the case of lipophilic metabolic efficiency. However, something I will notice is that when you start optimizing multiple properties and you start combining this is overkill. If you sum LLE and lipMAT, log D disappears. So why would you be acquiring log D at that point? All that you're left when you sum these two is the product of EC50 and clearance intrinsic. And this is what mechanistic ADME will tell you. If you look at those projections, given the assumption that you have good absorption, and this is a C average driven efficacy, what you're left with is the product of EC50 and clearance intrinsic. So if we get to the same conclusions, and that's first principle science, and that's what we use for clinical experiments, why wouldn't we use this earlier? And that's kind of what I'm getting to. How do we leverage this early on? For molecule making, this is very effective, I find, but even more effective for selecting what compounds go in vivo. If you treat your PK experiment, your rodent PK experiment, as a filter, this is, in my opinion, very inefficient. You're going to do more PK, and sometimes the PK in rat is going to be different from the PK in human because they have different enzymes. What I like is using mechanistic ADME to get expectations on what the PK should be in the rodent and in the human. And then the rodent experiment becomes a way to validate that expectation, and then you have a quantitative expectation for human. And if you do that, it's a way more objective way of prioritizing compounds. There's less discussions in meetings, and I find you end up testing less compounds and move more quickly, focus on in vitro properties that you get quicker, so your SAR is faster. And I'm going to provide examples of that. Okay, so what does this look like in practice? Let's say that you are thinking of a molecule, then you're just using in silico properties. Potency prediction, we don't have the best tools at this point, so you're probably going to work with assumptions. You're going to start from the lead molecule potency, and you're going to hope that you retain it, and you're going to calculate properties for the rest. But if this is a molecule you made, then you're going to have in vitro potency, you're going to have in vitro clearance intrinsic. That's most of it, really. The other properties you're going to calculate with machine learning tools, and you generally have good estimates, even for small companies that don't have large data sets. So you take all these input variables, you create PK curves, and then we talk about the PKPD assumption. If you think that the efficacy is driven by C mean or time over threshold, from that curve, you just extract the C mean, and you compare that value, you make it free, you compare that with the potency. You do this for all the compounds in your SAR table. You simulate the same dose. You simulate maybe 3 mg per kg in human. And that ratio between potency and C mean becomes your score. So you can rank order compounds based on this. And every week, you know if you have compounds that are more promising than your current lead, and those compounds you're going to acquire additional data for, or if they're not, you're going to drop them. Where do you get the models is another important question. I think solubility it's straightforward, not because it's easy to predict, but because even with in vitro data, it's not very well predicted anyways early on. The compounds that we make, the solid form is going to be different from the solid form that we're going to be using in the clinics. And that's going to have a huge impact on the solubility. The solubility that we measure early on, kinetic solubility, it's a precipitation assay with the MSO. It's very optimistic. Oftentimes, it's completely misleading. There's papers about that, very good papers from AstraZeneca about that. So I really like the solubility forecast index from Hill and Young. Essentially, it's the sum of log D and number of aromatic rings. I found when I did analysis, comparing that with the thermodynamic solubility of the most stable crystalline form, I found that that's as predictive as kinetic solubility. But you get it early on with simple physicochemical properties. When it comes to permeability, I'm going to comment on this BPI, Balanced Permeability Index. I have 3 slides on that. That's going to cover most of small molecules. If you have a particularly complex conformational effect, you can rely on molecular dynamics. There are good packages now out from commercial softwares. Volume distribution, I'm going to comment on the model from Korzekwa and Nagar. I think this is an incredibly fascinating and helpful tool. We did publish quite a bit on that model, but essentially you boil down free volume to affinity to tissues, and you can use microsome binding, which is an endpoint you're going to use for clearance anyways, or you can use log D and pKa to predict that LKL, that lipid constant of affinity. Clearance intrinsic for metabolic stability. I was astonished to find that even in a small company that doesn't have large data set, just with literature data, you can build very good models. I hope we can publish on that. I think we found a clever way to get them in terms of data sets. I really hope we can publish on that soon, but I know you can get good models for clearance intrinsic, even not having large data sets. When it comes to potency, you gotta cross your fingers. It's a guesstimate, it's a guess. It really is the name of the game. And it's something that we know something about through structure-based biology, but we have to accept that a lot of our hypotheses are not gonna work out. Okay, so Balanced Permeability Index, we stumbled on this thing. We didn't look for it. We're in a project where somehow log D and polar surface area were decoupled. And that was a first for me. Most of the times I've seen correlated polar surface area and log D. And this particular chemical matter differentiated these properties. And I knew from machine learning models that they're very important for permeability prediction, but I didn't know how to combine them quantitatively. But we found that the ratio of the two, while normalizing log D by size, was very predictive of permeability and efflux in our chemical series. So what we did, we applied that same simple formula to a Genentech dataset that we published at Genentech a few years back. And what's good about that dataset is that there is all permeability data from one lab, and there is experimental log D. So we're able to apply this concept of normalizing log D by size and polar surface area. We multiply it by 1,000 so that the numbers is closer to 1. It's not really meaningful, but we liked it that way. And even in this Genentech space, what you can see is that a BPI over 1 almost guarantees no efflux or no high efflux, which is red in the plot on the left. And a BPI less than 0.5 almost ensures that you have an efflux problem, and in between you have this toss coin, and it's different likelihood. You gotta accept the risk. Now, if you keep in mind those numbers, we move into the BMS space. Now this is thousands of measurements in Keiko2, different assay, different lab, and we're looking at efflux and we're looking at permeability. You have very similar trends with similar numbers, less than 0.5. You have efflux problems. Over 1.5, you don't. And we also looked at prediction of brain penetration in rodents. And surprisingly, there was a pretty astonishing correlation for these metrics. Something I should mention is that for the rodent experiment, we had experimental pore surface area from chromatographic experiments. That makes a big difference. But this is something really enabled decision-making early on for us, especially because for reasons that I don't want to go into, we were having troubles getting good measurements from the permeability assays because of recovery. So after I left BMS, they continued on this field, and they added this additional term that takes care of conformational effects in larger molecules like heterobifunctional degraders. And what they found is that this corrected BPI is predictive of absorption in rodent, which is quite astonishing because even in vitro assays are not predictive of absorption in rodents when it comes to heterobifunctional degraders. Assays really fail in that space. So sometimes simple concepts, they can go a long way. So I think we have tools for permeability. I guess that's what I'm saying. When it comes to volume and distribution, this Korsakoff-Nagar model, I really like how it breaks down distribution in three different phenomena. One is compounding plasma. That's constant. That's the plasma volume. Then there is the intracellular water. You see a lot of letters and symbols. Those are all tabulated values. The only measurement in that intracellular water space is plasma protein binding, because obviously in intracellular water, you have albumin and other plasmatic proteins. And then there is the lipid compartment. Lipid compartment, again, volume tissue, which is tabulated, plasma protein binding, which you need for clearance anyways, and you have in intracellular water. And then you have this LKL. LKL is affinity to lipids. You can measure the volume in rodent and back calculate LKL. You can derive LKL from microsome binding, and that's where the novelty came from the Korsakoff and Agar approach. They fitted LKL with microsome binding measurement, microsome binding we need for clearance, and it's unsorted lipids. So it's a very good in vitro system, and it's working very, very well. And we, in follow-up papers, we show that this works well, not just for human, but also for preclinical species. And actually, you can derive LKL also from log-D and pKa. So you can look at the papers, but in our hands, the volume and distribution predictions from in vitro numbers is comparable in accuracy with predictions from cyno. And that's much cheaper. So how do you apply this in decision making? I thought of a comparison between efficiency matrices and mechanistic modeling, and I think half-life provides one, because essentially you can get volume from Korsakoff and Agar, and you can get clearance from the Western model. So from 3 endpoints, you can get a half-life prediction. And I look for what do I compare this with? And I think LibMeti is a good comparison, because with LibMeti we try to optimize half-life. This is something that we talked about in a paper a few years back. We provided statistics showing that when you manage to decrease clearance while not decreasing lipophilicity, you have a better chances of increasing half-life in rodent. And therefore, we suggested that LibMeti could be a good approach. And an example of that is given by this Amgen dataset on the right for these 3.1 inhibitors, where essentially they knew that the site of metabolism was at the benzylic position. When they tried to change the central ring, didn't change, lipophilicity didn't change, the site of metabolism, half-life is the same, clearance is the same, volume is the same. As they modulated the left side, so far from the site of metabolism, but they decrease in lipophilicity significantly, clearance goes down, but volume goes down, and half-life is not really improved. But when they change the metabolic soft spot with the lipophilic moiety, then they do not damage volume, they improve clearance, and that brings a substantial extension of half-life. Okay, so the concept is, don't just think of fixing clearance, think of fixing clearance without losing volume. The Roche people in Basel, former colleagues, they brought this concept on with this analysis on LibMeti and half-life in human on their clinical candidates, and in preclinical species, they showed that there is some level of correlation. So on this slide, I am comparing on the right LibMeti as a predictor of MRT. If you look at the paper, we actually have statistics, we have R-squares, but I think this is better for visual. you talk about half-life, what you're really talking about is the ratio of volume and clearance, which we call MRT, mean residence time. Shorter MRT is in red, longer MRT is in green in this plot. If you look at LibMT, you see that, yes, you have increasing ability to enrich for better MRT through LibMT binning, but on the same inputs, except for maybe PK, I think we also have PKA on the plot on the left, which is using mechanistic equations. But other than that, it's the same input. You have a much better ability to predict and enrich presence of longer MRT. So the point is, same input, different approach. The approach on the left is mechanistic. It's giving us qualitatively better answers to our questions. So why wouldn't we use that? For people that are passionate about PBPK, I am. PBPK is a field that has been developed thinking of clinical experiments. At that time, we have a lot of data. So you can use these tissue partitioning models from Malcolm Rowland or from Patrick Poulin or from Vieira. And you can derive from log P, PKA, log D, blood-to-blood partitioning, and other in vitro endpoints. You can derive the partition at the individual tissue level, which is what you need to create a PBPK curve, which is a bunch of differential equations describing flow across different tissues. But if you want to apply that early on in discovery, you don't have all that data. So what we did in this paper from 2 years ago, we simplified the approach. We said, okay, let's look at correlation between different tissue compositions and KPs. And we derived essentially a tabulated values for interrelationship between KPs. Most tissues behave like muscles, and some tissues behave like liver and kidney. So if from my volume, I remove the plasma, I have a volume, the tissue volume value. Now, simply between these interrelationships, I'm able to derive immediately the KP across all the organs. So if you have volume from the animal or from FUMIC and plasma protein binding, you can get immediately all the KPs. And what I'm showing in this slide is that simply between plasma protein binding, microsome binding, and microsomal clearance, you get projection of clinical PK for verapamil, midazolam, and warfarin, the IV portion of the clinical PK, that is actually very, very quantitative. Okay, so you can apply PBPK quantitatively early on with limited data. Okay, so let's see this in action. I'm looking at two projects from BMS. We published this data, Project 1 and Project 2. Project 1 is the one that we used to develop the method. And in Project 1, we're assuming that time over threshold, so C mean, will drive efficacy. So with our PK projections, we extract all of these C means and we compare that to the potency. And we get this C mean score, and we quantify this C mean score for all the compounds in the projects. And then we stratify them by a chronological splits. So we have the first third that we made, and the second third, and the third third. And within each split, we have compounds that were characterized in PK and compounds that were not characterized in PK. The one that went for PK are in yellow. So what you see is that over time, the score improves, reflecting the fact that we are making progress in the project. And at any given time, the compounds that went in PK look better than the ones that didn't go for PK. I mean, showing that the decision-making tool here reflects the decisions made by the humans. The pink box is the compounds that were shortlisted for the clinical experiment. So the 5 compounds that we were considering for human, they have much better scores compared to the other compounds. And especially if you focus on the plot in the middle, when we are also accounting for Cmax and hERG IC50, and we are building this therapeutic index, the compounds that we're considering for the clinical experiments are very much differentiated from all the other ones. We didn't use this tool to make decisions, but we were able, with this first principle tool that knew nothing about the project, to get calculations in minutes. And these calculations reflected exactly what humans took months to come up with. The other point is that after we saw this, for the backup project, we started using this approach, and the amount of PK we were running decreased drastically, simply because not many compounds were better than the current leads. And that focused the optimization on in vitro properties, because we realized from in vitro properties, we can tell most of what we need to know. That means you do SAR on data that you get faster. Project 2, we don't have the split for PK. We didn't have that in a database. It's an older legacy project. But again, the score could recapitulate the progress over time and the fact that the clinical candidates look better than the other molecules. So if you like statistics, you can essentially calculate the ability of fishing compounds that you're considering for clinical experiments. You will get an AUC of 0.97, which is pretty remarkable. And if you do the same for fishing compounds that you consider for your PK experiment, then you'll get an AUC of 0.86. I wonder how many of those decisions are optimal, given that we make them in real time in projects, and sometimes maybe that 0.86 reflects problems with the algorithm, but also maybe problems with decision making. So all in all, I think in my hands, this significantly decreased animal testing, improves the efficiency of decision making. The meetings are way shorter because this is an objective metrics. Of course, if you have hypotheses that you're testing, you should add those compounds. But sometimes I feel that when you make decisions based on PK slots, we have this many slots, you end up making suboptimal decisions, and this is a correction for that, I think. I've seen it applied to many other projects after this, and it consistently identified the best compounds, unless there was a lack of in vitro and vivo correlations, which I would argue is a problem for any other MPO. So all in all, you can use different modeling approaches as tools in your toolbox, IVAVC, PBPK, machine learning models, and you can leverage them in an objective multi-parameter optimization function, provided that you have established good IVAVC. We made this possible by simplifying a lot of the PBPK science and making it possible with less data. And in our hands, this works really well. And even with in companies that don't have large data set, you can get good machine learning models to support this. Of course, the limitation is that we can't predict potency accurately at this point. Sometimes we can, but we don't know a priori. For some targets, free energy perturbations work amazingly, but for many, they don't. And so I think that when we see more progress in that space, combining that with this, it'd be an incredible efficiency gain for the field, but for now, that's a holy grail. Property-based design, I think that's very powerful to enable us to have good ideas. Beyond that point, I would suggest using more sophisticated approaches to scoring, but I don't want to underestimate the value of harnessing chemical intuition, because that's where most of the best ideas and most of the crucial moments for a project come from. With that, a lot of people I need to acknowledge for the work. They are in the publications. They were amazing collaborators across different companies. I want to thank different companies for enabling the research that I presented today. And with that, I think I'm ready to take questions.
Amazing, Fabio. Yeah, that was such a great presentation. Yeah, and thank you for, showing us kind of the holy grail and, initially guiding us through, what challenges were earlier, how they were overcome, and really how the field developed and how all these models are connected with each other. So really, really helpful presentation. Thank you, Fabio. We have some questions here by the audience already. So maybe let's just get started there. One question comes from Xiangyan Yi, and he is wondering, or they are wondering, I should say, for small molecules in silico models are quite precise and show a good correlation with in vitro data. However, larger molecules like proteins, peptides, and oligonucleotides, to my understanding, is that large pharma still heavily relies on in vivo DMPK data over in vitro ADME assays or predictions. I'm curious, are there any emerging models specifically designed to predict the ADME profiles of those big molecules?
That's an excellent question. I think before getting to the models that predict, we may have isolated examples, but I think where the biggest gap is actually the mechanistic understanding of what's happening. Once we have that, then we will be able to have bespoke in vitro assays, and we'll be able to develop models for the bespoke in vitro assays. What I fear is models predicting the wrong thing. And I've seen it a lot. So I think for a lot of these different modalities, we are back 20, 30 years ago compared to small molecules. We don't understand what's happening. If you take oligos, we don't understand the subcellular distribution. We don't understand the endosomal escape. We don't understand any of that. We don't have assays to predict that accurately. If you take products, the problem there is that most of our PBPK assumptions rely on the idea that compounds move freely between different compartments of the tissue, and that's fast. And the elimination from plasma is the slower process. But for products, we see products degraders. I know there's different terms. For heterobifunctional degraders, we see that the KP, so the plasma to tissue ratio, changes over time. What that tells me is that the compound doesn't go immediately back to plasma once it's eliminated from plasma. At that point, all the assumptions that we use in our PBPK models, in the Western model, in the polymer distribution models, they are all not valid. So at that point, yes, maybe we can develop in silico models, but what's more pressing is developing an understanding of what's happening and in vitro assays that are guiding us there. In absence of that, you're correct that we rely on preclinical PK heavily. So that's an area where I think if I were a freshly graduate person, I think I would focus on, because there is a lot to innovate there.
Awesome. Awesome. Yeah, no, that's super interesting. Yeah, I didn't know that, KPUU was time dependent with some of those degraders. And maybe larger molecules. That's super, super interesting.
Maybe lysosomal trapping, right? We don't know.
Yeah, interesting. Yeah, definitely important to really develop the assays that, help us understand, those complex molecules. And those may be, as you pointed out, different from the assays that we use today to, to kind of prioritize small molecules. Another question here comes from Ann Tran from Amgen. She's wondering, could you please comment on building models on chemical space where non-hepatic clearance is the main clearance mechanism?
Okay, well, that's another excellent question. I like that the audience is getting me on all the exceptions. So when it comes to small molecules that are already bioavailable, there is an assumption that you have enough permeability to make it to the gut. And that's usually sufficient permeability to make sure that the compound is not easily reabsorbed from kidneys or bile. And therefore, the most prominent mechanism is metabolic elimination. There are exceptions. I was at the ISSX conference 2 weeks ago or last week, and there was a lot of talking about intestinal secretion. And intestinal secretion, it's a phenomenon that seems to be happening. It seems to be PGP enabled. It seems to be happening more so for compounds that are moderately permeable and lipophilic. In that space, we don't have good models. We also don't have an understanding of what we see in rat, will that translate into human. But what I do know is that if you have high in vitro clearance, that's going to be a problem anyways. So in those cases, you want to do mechanistic ADME studies in vivo to understand what's happening on your leads. But at the same time, you're going to get a projection just looking at metabolic clearance that it's your best case scenario. So if your best case scenario doesn't look good, you know that that's not a molecule you want to pursue anyways. So you're going to have to caveat that a little bit more. But I still think that there is validity in the approaches that I described for that type of molecules. I hope that answers the question. In fact, some of the things, it's very interesting, intestinal secretion. It's something that now people are paying attention to, because really what you need to do is have mass balance studies already in IV, which is rarely been done for clinical candidates. But there is an understanding that probably this is an elimination mechanism that we have overlooked for decades, and many marketed drugs may have that.
Interesting. Yeah, no, and super cool. Yeah. Next question here comes from Tudor Oprea.
Oh.
And the question is, if you were to pick 3 to 5 properties that are, semi or even orthogonal to cover the ADME property space, which properties would you choose? So, he's basically there wondering, where would you want to get more data and which properties?
Well, Curated intrinsic dominates those projection and requires an enzymatic process, which means there is a recognition with the receptor. Now, these are very promiscuous receptors, so oftentimes the binding aspect is not as prominent as it would be for potency, but it's somewhat important. So that's a property that we can kind of predict, but because it has a very important weight on those projection and because it's not as predictable as something like permeability, it's definitely one that we want. If you measure Kern's intrinsic, you're gonna need the binding in incubation. Otherwise, an in vitro number is not reliable. And that you can use for polymer distribution as we described. So microsomal binding will be my second one. That you describe the IV component of your curve. Permeability is very important. If compounds are not permeable, they're not absorbed. I would argue solubility, but as I said, we don't have good solubility measurements early on. We just don't. It's something that took me a while to appreciate, but kinetic solubility is better than nothing, but it's really not a great predictor of thermodynamic solubility of the most stable form. So I wouldn't give as much importance to solubility, but I might acquire log D because I can get a proxy for solubility through that, and I'm able to assess improvements in all the other properties through that. So we are at 4, and the 5th would be potency, of course.
Awesome. Awesome. Yeah, no, that makes a lot of sense. I think what you said about the form is like so eye-opening that, of course, you're not going to have like this amorphous form that you initially synthesized with like 2 milligrams or maybe 10 milligrams of compound. That's not going to be your clinical form. And solubility, I've also seen that very, very, very dramatically.
Yeah, and you tell me because you're a chemist, but my understanding is that with the current purification methods, with the current high throughput purification methods, most of the times compound is going to be amorphous, or way more often than it used to be in the past, and that can really fool you because you think the compound is soluble when it isn't. There is some information in that though, because if a compound is soluble in its amorphous form, it indicates that even if it is insoluble for the crystalline form, there could be room for spray dry dispersion to help the formulation. So I'm not completely disregarding the value of that assay. I'm just saying you will have to do a lot of formulation work.
Yeah, makes total sense. Makes total sense. Yeah, we're quickly coming up on the top of the hour, but I think maybe a couple of more questions here. So another question comes from Alexei Gerasiuto from Schrödinger. Interesting observation that even small amounts of ADME and DMPK data can allow for predictive machine learning model generation. What would be the reason for that? Is it maybe staying within a narrow chemical space on a specific program, or are there other reasons?
I didn't say that it would take small data sets. I said that it would take that even without owning proprietary data sets, you can. And I hope I'll be able to say more about that at some point. But the answer is not a small data set for sure.
Okay, makes sense. Yeah, thank you for clarifying that.
I know I'm being enigmatic. I hope I'll be able to say more in the future.
We're looking forward to hearing that. Maybe on another Drug Hunter Flash talk. Maybe last question, since we're coming up on the top of the hour. Last question is from Yoshi Ishihara. Could you compare the predictability of ADME models trained on in-house data, for example, at BMS, versus ADME models trained on broader literature data?
I think that if you have experimental data sets from large companies, they're going to outperform everything else that is out there. And actually, there is a very interesting publication from my former group. Joe Napoli, I think, is the first author. And he's looking at combining Roche data and Genentech data and see how that changes the predictivity. I think the answer to your question is going to be in that you will see that more data, broader chemical space improves models. With that being said, if you look at models that are almost as good as the assays, and you compare that with decision making based on chemical intuition, there is a space in between where literature data sets can play and improve efficiency within certain caveats and assumptions. I do think that, and that's something that is underestimated, modern approaches to machine learning, like a graph neural network, they have improved, generally speaking, the predictivity a little bit, not terribly, just a little bit, but they don't do a good job at quantifying uncertainty. But approaches like extremely randomized trees, which have this voting system, if you may, you can take the standard deviation of this voting system. You do get a sense of how solid the prediction is from that standard deviation. So even for literature data sets, if you couple the prediction with the uncertainty, you'll be at least able to tell which predictions you should trust and which predictions you shouldn't.
That's very, very helpful. And yeah, a very nice, I guess, closing statement here, Fabio. So yeah, I want to thank you again for an amazing presentation, for sticking around to answer the audience questions. We had many more, unfortunately, that we wouldn't be able to get to for time reasons. But yeah, we'll also want to thank the audience, of course, for dialing in today and asking all these thoughtful questions. We really appreciate your engagement. And yeah, again, round of applause for Fabio, I would say. Virtual round of applause is coming in. And we will be here next week again with Peter Skoos-Cox from Novartis, and we will be talking about AI meets plate-based imaging. And we are going to harness hallucinations for high-content screening next week. So stay tuned for that. Very interesting topic.
Thank you very much. Have a great day.
Thank you, Fabio. All right, everyone. Take care. Bye for now.